am very happy to introduce this week's speaker, Dr. Sheila Kitchen. Sheila Kitchen comes from Oregon State University, where she did her PhD under Virginia Weiss. From there, she went back to the East Coast to do a postdoc at Penn State under Ileana Bombs, where she did population genetics on reef corals. She then moved back to the West Coast to work at Caltech, learn a different perspective with Dr. Joe Parker, where she worked on evolution of species interactions in the insect realm. She is now an assistant professor at Texas A&M University in Galveston. I've known Sheila predating all of that from when we were both master's students at UNC Wilmington. And since then, we've always sort of revolved around the same field of ecology. So once she moved down here, thought it was prime opportunity to invite her for a talk. So today she'll be sharing some of her work on population genetics of reef corals. And with that.
Thanks, Kenny, or Dr. Hoadley, sorry, reverting back to our old days. So I would like to thank everyone for coming today. This looks like a pretty sizable crowd, and so that must mean you're really excited about corals, and I'll try not to depress you too much. So I tried to make it more positive, sources of novel genetic variation in endangered corals and their symbionts, and maybe we can glean some hope from what I'll show today, because I know a lot of the coral literature right now and a lot of the coral discourse is not so positive. Okay, so as Kenny said, I am an assistant professor, just started in January. So my lab, oh, thank you. Okay, so my lab is just up and coming, not a lot of projects. So I'm going to talk a little bit about old stuff today and then how we're moving that forward. And I'm trying to slide advance, but there we go. Okay. All right, so what motivates my research is the fact that organisms, us, all the way down to single protists, are really communities of symbionts. And these symbionts can influence our behavior, they can influence our development, and they, of course, influence our health. Interactions between these hosts and their symbionts are mediated by complex chemical, behavioral, and molecular signals. So from our social insects like the rove beetle grooming its ant host, I had to put that in there for myself, to the endosymbiotic algae that live inside corals. These associations can be influenced, or sorry, these associations can influence both the genome structure and the function of both partners. So much of my research has revolved around the question, how does symbiosis evolve? But really specifically what I'm trying to get at is what is the degree of which these symbiotic partners can acclimate and or adapt to a changing environment. Okay, so here my research program is kind of at the interface of basic questions on symbiosis. So how do these partnerships form? How do they break down? And what keeps them going? And then I started with this postdoc at Penn State to really think about genomics and how we can start intertwining that into these questions on symbiosis. So can we add in the host and symbiont genetics and genomics to these interactions? And then of course, how does the environment structure all of this and alter these partnerships? So some things we're currently thinking about, or what I'm going to talk a lot about today, is how can hybrids act as novel habitats for symbiont evolution? I'm also still really interested in host-symbiont partner specificity and fidelity, and I use model systems for this. I'm starting to develop some genetic tools like RNAi to probe the functions of these cnidarian genes in these model systems. And I've also been helping develop genetic tools for coral restoration, and I'll talk about that briefly. So the first little vignette today is going to be on this hybrid as a novel habitat. Second, I will just briefly touch on this tool that I developed that's being actually implemented right now in coral restoration. And then third, I'll end with this kind of study that came from an odd sort of spawning event where we saw spontaneous development in a single colony, otherwise known as selfing, and we were able to track mutations and those offspring. So I'll talk about that in a bit. Okay, so we'll start with hybrids. Okay, so this audience, you don't need an introduction to corals. I'm sure you've heard all about it. But why do we really care about the coral reefs? Well, they provide extreme biodiversity. You've probably heard the metric that they provide 25% of the biodiversity on the planet. And really the corals themselves are the foundation, right? They build the structure, the complex habitat that supports this biodiversity. The corals themselves act as microhabitats for a bunch of microorganisms. So notably the algal symbiont, this is its key home. And this nutritional symbiosis between the algal symbiont that lives in the coral is key to its survival, right? Because these are otherwise nutrient poor environments. And this ecosystem provides us humans things we care about, like shoreline protection, nurseries for juvenile fishes that then end up in managed fisheries, a bunch of jobs for people who live on the coast. So people like us care about that sometimes. And also it provides wealth of services to indigenous communities who've been using these basically forever. But as I said, we've gotten a sort of a doom and gloom story about corals recently because they are severely impacted by climate change. Obviously, here you're seeing the response of coral bleaching, which is when the symbiont is expelled from the host and that loss of pigmentation gives this white appearance. And so you see this bleached coral. So the frequency with which these thermal events are now occurring has actually led to significant declines in coral populations with species extinctions predicted in the next couple decades. So that's not great. All right, so little coral biology again for this audience, probably not necessary. If you were to look closely at a coral, so we'll zoom in, you'll see it's composed of a bunch of polyps. And each of these is a genetically similar, it's the same genotype as its neighbor. And if you look closer at this tissue, there's two tissue layers. I give this background because some people don't know where the Symbidiniaceae or the dinoflagellates, or as I've been calling them, the photosymbionts live. And so they live in this gastrodermal tissue inside their intracellular inside of the coral cells. And this relationship is really built on an exchange of products. So photosynthate from the symbiont then gets exchanged to the host, and in return, the host provides its own waste products to promote that photosynthesis. And so how these symbionts are acquired is kind of important to part of my story today. There's two different ways in which you can get your symbiont if you're a coral. You can be a broadcast spawner, which means you release your gametes out into the water column. And then that process generally means that you do not come with your symbionts, but you must get them from the environment. So that's a horizontal way. And this is the primary way in which corals acquire their symbionts. The other way is brooding, in which they directly transfer from the parent colony to the offspring, and brooding meaning that these are fully formed little larvae that swim away. So that's internal fertilization. So when we think about broadcast spawning corals, they have a lot of similarities to plants. So just play along for a second. I promise there'll be a point to this. Okay. So they both rely on external fertilization, right? And that introduces many opportunities to have two species that wouldn't normally come in contact. Their gametes can come in contact because of this process of external fertilization. So while hybridization has been considered this significant role in shaping plant evolution, we think it's a really positive thing. And animals is generally considered to be a bad thing that is just there to reinforce the species boundaries. Another thing that they have in common, another thing they have in common is this passive dispersal, right? So the seeds get, they fly away, and then that's how we get new plants. And in coral larvae, they swim away, they find suitable habitat, and they settle. And so just like plants, this can lead to differential recruitment over their geographic range. And so this leads to this process of reticulate evolution, where you have this web of secondary contacts between species, such that the variation within a single species sometimes becomes indistinguishable from the variation among similar species because of these secondary contacts. And this process makes it very hard to understand the history of corals. And that's why we have all these phylogenetic disagreements. So today I'm just going to focus on the genus Acropora. And while that seems like I'm narrowing down, there's still a lot of species within this genus. It's highly diverse. They're broadcast spawning corals, so they have this possibility of hybridizing. Most of the diversity is in the Indo-Pacific with about 100 species. In the Caribbean, we're pretty lucky that we have 2. That makes it much easier to study. And there's only these 2 sympatric species. So you've probably heard of the elkhorn coral, Acropora palmata, and the staghorn coral, Acropora cervicornis, for their distinct morphologies. So they're easy to follow based on that. They have undergone dramatic population declines. Here, the Pleistocene on the bottom x-axis is on the left, up to around 2011. And you can see that the occurrences of these field surveys, they plummeted. So not only are they not really present anymore, they're no longer dominant either in those particular locations. And this decline started before we saw the bleaching events and we saw the disease events, which is that white man disease. So really the decline was because of human activity, anthropogenic sources. And because of this, they have been listed as these endangered species, and they're active targets for coral restoration Caribbean-wide. Okay. They also form one of the only recognized hybrids in corals. So there's a lot of hybrids in corals. There's a lot of cryptic speciation. We're still trying to figure it out, but this was the only one that was ever given a name, because up until 20 years ago, we actually thought it was a third species. And then with the advancement of markers, molecular markers, we discovered that not only was it not a species, but it was classified as an F1, a first generation hybrid, and it was called the mules of the sea, effectively a dead end. But what we are seeing in the field surveys is that their abundance is starting to increase at a faster rate than their parents. And so that begs the question, is there something else happening here? Is that just a product of asexual fragmentation that corals are famous for? Or is this really through some new sexual activity between the hybrids? So another little tidbit about the hybrids, they can outperform their parent species. So first, hybrids dominate the shallows. These are the high light, high temperature environments, and they thrive in those environments. They overlap with Palmata, so that was the elkhorn species, and then Cervicornis tends to dominate in the deep. So there is some stratification, but not much for the hybrids and Palmata. They also seem to be less impacted, although this is very preliminary data. Oh yeah, I have to show you it. Okay, there we go. So they seem to be less impacted by bleaching and disease. Again, very preliminary, needing more data on that. But this suggests that they have some hybrid vigor, meaning that they can outcompete because of the unique genetics in those environments that the parental species do not have. So we kind of return to this question of, is this really an evolutionary dead end? Is there no adaptive potential in these hybrids, and so we shouldn't care about them? Or do they offer an opportunity to move adaptive alleles into the parental species, which are currently not helping themselves and undergoing significant population declines and enhancing genetic diversity in the parental species? So a little bit more on what we do know. If we look at the reproductive barriers, I did show you that there is some slight habitat isolation, but not much in the sense of like where gametes can go. And they're going to float to the top and make contact with each other. There's also some temporal, isolation. They have about a 2-hour gap in their spawning time. So not a really big one, but there's a little bit of a gap. They also have some evidence of gametic incompatibility, meaning that the eggs of Palmata are not receptive to Cervicornis, but Cervicornis is receptive to Palmata sperm. So we see some level of asymmetrical gene flow, potentially. And I've lost it, sorry. Or there. Okay, so post-zygotic, we know that they're viable because we see them out on the reef. So these hybrids, at least at the F1 generation, they live, they don't die. And we've been able to actually make them in the lab. So we know that it's a really grainy video, but they're swimming around. This is what the fieldwork looks like for me, which is like trying to get my camera to on my phone to get a video. It's not the best, I need help. So with that, we know that they're everywhere. So hybrid viability doesn't seem to be an issue, at least at the first generation. But they may be sterile. And that we don't have any good information on yet. So hybrids have been observed spawning in the field, but because they're not a direct target of restoration, we're not actually collecting gametes from them. And so no one's actually, as far as I know, tried to make later generation hybrids from these hybrids that have spawned. So that's just a lack of effort, but could be reality that they just are sterile. And then the other thing that could happen is hybrid breakdown. So this is when the hybrids are fertile, so they make their gametes, they try to combine those two genomes, but it breaks down later. So they have their babies, the babies swim, they recruit to the reef, and then for some reason they have lower fitness because of the combination of these two genomes that just really aren't compatible with each other. And so this reduces the fitness and leads to hybrid breakdown. So we have evidence that these hybrids can make back crosses. So that means that the hybrid can reproduce with cervicornis, but so far we don't have any evidence of them doing that with palmata, and we have no evidence of these later generation hybrids. So given that the hybridization, at least from the surveys in the field, we're seeing more hybrids in the field, understanding the extent to which these hybrids can mate is an important piece in determining how we can use hybrids in a restoration context, because if they are starting to do these things, we have to be sort of careful about how we apply the hybrids. But we also can think about how it might shape the adaptation of these species. Okay, so questions we had going into this was there a recent breach to the hybrid sterility barrier, meaning that the hybrids maybe before weren't able to have viable offspring, but now they can? So that's a question we wanted to ask. And is there differential introgression across the hybrid genomes that would give us some sense of whether there's incompatibilities between the two species? To get at this first, we had to get some genomes. So here I'm just showing you the Acropora palmata chromosome resolved genome. And it's resolved into 14 pieces, and it's beautiful and it's great. At the same time, I did assemble the cervicornis genome. And Nick up there in the corner was recently able to, like as of 3 months ago, make a linkage map and resolve that as well into chromosome resolved. So what you're seeing here is the palmata chromosomes on the top, cervicornis on the bottom, and any place where those colors cross means that there's some place where that piece of the genome from palmata is not in the same position as it is in cervicornis. So by and large, we don't see large rearrangements. We see little rearrangements between the genomes. If you look at this at a whole genome level, there's about 3.5% that differs between the two species. And that might not seem like a lot, but if we put it into context of humans and chimpanzees, we're about 1.2%. So it's actually quite different, even though there weren't large scale rearrangements. Oh, okay. To get at this question, we also needed more sequencing than just the genomes. So we targeted the 3 different taxa across multiple hybridizing zones. So for example, Belize, USVI, Curacao, and there was a couple hybrids in Florida. And we took advantage of different types of ways in which we could collect that data. And from all of this, we found what I'm going to show you most of the results on today is from the genome resequencing data. And we've had about 500,000 high quality SNPs that we could distinguish taxa, populations, and individuals. So one of our first big results was that hybridization is bidirectional. So I'll walk you through this a little bit if you've not seen one of these plots before. This is an admixture plot. Each one of these bars represents a sample, and the color indicates the population that it was assigned to. So in this case, we're looking at just a K of 2. If you are in the yellow population, you are cervicornis. If you are in the pink population, you are palmata. And if you are in the middle, you get a mixture, right? So you get a little bit of pink and you get a little bit of yellow. And in fact, the majority of the hybrids that we did whole genome sequencing on were F1 hybrids. Despite the fact that we tried to find complex hybrids, they all came out to be almost F1s. But what was really exciting is we actually found backcross palmatas, and that was counter to the previous results from prior work that said there was only cervicornis backcrossing. So now we can actually say that this goes in both directions. It's not just in a single direction. The difference between our work and the previous work is that that was in the Mesoamerican reef track. And so we think that the frequency and direction can vary quite a bit during these spawning events, and also maybe across these hybridizing zones in which they're more receptive to one backcrossing or another. Okay, so the next kind of result that really had us puzzled for a long time, and probably why this hasn't been published yet, because we're still trying to wrap this up. So in our naive expectation, right, we just saw that an F1 was like 50-50. So that means it should get 1 chromosome from palmata and 1 chromosome from cervicornis. So this is what our expectation of what a F1 hybrid would look like if we looked at the whole genome. But we dug in a little bit further because We really were hoping we would find F2s, and we were just looking for some patterns that would tell us that they weren't what we thought. So this is a complex plot. So I was preparing you for that with my delay. Okay. So on the bottom, on the X is the genomic position across chromosome 1. On the Y is the allele dosage. So the way to read this plot is if you are at 1, you have an allele from both parents, right? So you're heterozygous across those alleles. If your alleles jump up to 2, that means that you're homozygous for palmata or homozygous for cervicornis. So you have 2 copies of that allele from that particular parent. So what this suggested to us is that there's these stretches where the F1s are no longer looking like F1s. They have stretches where they've lost that heterozygosity and they're homozygous matching one of the parents. And so that's really interesting and very confusing. So we went and we counted just the number of tracks across here. And the other thing that we found when we started looking at the numbers is not only do we find a lot of these across the genome, so this is across all of the chromosomes, not just chromosome 1. So we find many, and we find that there's a bias towards palmata. So that means that if you were to look at any one of these random stretches of loss of heterozygosity, it's most likely going to be palmata. So the palmata genome is being favored when it fills this gap. So that's interesting. We also use a different approach. This is a window, a sliding window tree approach where we built phylogenies across our SNPs. And we also see the same thing where there's this bias in palmata hybrid relationship versus cervicornis hybrid relationship. So we tried to get at this as many ways as possible. And we also even looked at larvae that we made in the lab. We knew were F1s, and they also have them. So this isn't even like they matured and these happened, but this is happening from the conception. Yeah, so these results may indicate that there is differential introgression, that there seems to be a preference towards one of the subgenomes, and that the loci within these regions may actually represent those genomic conflicts that we were looking for. So this is just an example of one of those. I don't have this pointer. Okay. So I just pulled out one example here where we see that. So down here at the bottom, if it's a light blue color, that means it's homozygous cervicornis. If it's like a light gray, that means it's homozygous palmata. If it's dark blue, it means it's heterozygous. It has one of each. If you look at the top 2 hybrids there, they mostly look like they're cervicornis across that stretch. And this gene down here is centromere protein C. This is a core protein that actually is involved in connecting the kinetochore to the centrosomes, which is a key process in mitosis. So one thing that we were starting to see as we looked at the list of genes that fall in these loss of heterozygosity regions is many of them relate to chromosome segregation machinery. And maybe one of the reasons we don't see later generation hybrids is just because the chromosomes can't pair correctly and they can't segregate correctly during mitosis or meiosis, and that would lead to a hybrid breakdown situation. We also had differential expression data to look at just to see if there was something more we could dig out from this data. I'm going to tell you we could cluster these. There wasn't actually that many differentially expressed genes between the 3 taxa. This was from a common garden experiment. They were put in a common garden for a year to try to remove any particular locally adapted patterns. And sometimes you do see these cases where the F1 is going to be more like cervicornis, so AC dominant or AP dominant, meaning that it matches the expression of one parent, but not being intermediate to the 2 parents. And all I want you to take away from this is we found a lot of genes involved in DNA repair. Why do we care about those? Those tend to lead to these loss of heterozygosity regions and other organisms. So we think that these are really good candidates to follow up with in future studies to look and see exactly what's going on. Okay, so these potential mechanisms for parentally biased loss of heterozygosity regions, we didn't see large structural rearrangements. That could be one, but we didn't see evidence of that. I didn't show you this evidence, but we did look for hemizygosity, meaning that they had lost a chromosome, and maybe that's why we see this bias, but that didn't happen. They are diploid. But we are seeing some indications that maybe mitotic recombination differences are at play. We did see cases of gene conversion. At least transcriptionally, they have differences in their DNA repair. And we also see some potential issues with the chromosome segregation. So our data kind of hints that the reason we don't see later generation hybrids and we weren't able to capture them is because of this chromosomal segregation and these issues with mitosis. So now I'm going to pivot to a different story that came from the same data set, but then looked at this question a little bit differently. So this work was done by Hannah Reich. She was a grad student, now an assistant professor, so it's been some time. And she joined the lab when I joined the lab, and so I was like, I'll do the host, you do the symbiont, we're going to make this work. So she basically pulled out what I would call, or what we've affectionately called, the reeds out of the rubbish bin, and tried to make some story out of it. So the reason we could do that is all three taxa here associate with the same species of symbiont, Symbiontium fiti. And so that allows us to ask a question of how this symbiont is evolving within its host because it's found in all three. Symbiontiaceae, I'm sure you've heard that before. It's a complex family. We used to think it was one species. Obviously, it's a lot more than that. And they have symbiotic lifestyles and free living lifestyles. The main thing to take away, at least from my perspective, when we think about genomes, is they have really large genomes, about 2 to 12 times the size of a host genome. So when you do these sequencing projects, you're going to get it even if you don't want it. So that's fun. But we could take advantage of that. I was able to assemble a partial genome assembly for Symbiontium fiti, and then using the same sort of analysis pipeline, we were able to call single nucleotide variants, of which we did some downstream analysis. So a little bit more on the association here between Acropora, the Caribbean acroporids, and Symbiontium fiti. We know that sexual recombination is very rare, and the majority of the way in which the symbiont reproduces is through asexual fission. And so we can imagine that also Symbiontium fiti is an obligate symbiont, and the reason we think this is we haven't been able to culture it yet, like some other coral symbionts. And so given that, we expect that they're going to have high mutation rates, high genetic drift, and very low effective population size. Another thing we can consider is that they have 30 generations for every host generation. So that's a lot of generations of difference, especially for a species of coral that we estimate some of the colonies have been there for thousands of years. In Palmata specifically, we know that these associations are long-lived and they never switch their symbionts, or very rarely switch their symbionts. And I was just talking to Kenny, and there's a case of a restoration group where if you put them back in their native environment, you take them out of the restoration context, they switch back. So they're always there. And so, yeah, that's a really interesting thing about how faithful this association is. And we also know that the gene flow is really limited at much smaller spatial scales than the host. So we think that dispersal is quite rare. So that allows us to ask some really interesting questions. So one of the first questions was, is there subspecies divergence in the symbiont by host taxa? So you can imagine either there's going to be the structuring by region. So is geography the main way in which they're getting structured? Or maybe geography and then species, like host taxa? That could be a way that it happens. Or is it really structuring mostly by species and then geography? And that would have some different implications. And then do the hybrid host associate with a specific symbiont subspecies if that's the case? So what we found, and this is, I'm throwing a lot of data up at you at once. This is a phylogenetic tree. Over here in the yellow is Acropora cervicornis, and it actually creates its own distinct and highly supported clade of Symbiontium fiti that is different than Palmata and the hybrids. And so that was a little bit surprising. The other thing that we found was 12% of the genomic variation attributed to the host taxa, but only 2% was attributed to geography, which was really bizarre and not what we expected. And so what we think is happening here is that there's some level of disruptive selection going on that's kind of pushing these two, what we think are subspecies away from each other. And it's hard when you start talking about symbionts because then you get into like strains and clones. other things, but we'll call them subspecies for the point of this. So these Esphitae of cervicornis seem to be distinct from the others. And that kind of got us to this, like maybe the hybrids really are this novel environment of which the symbionts are living, and maybe that has some structuring effect. So we proposed two different ways in which this could happen. You could have evolution within a host, right? So as the larvae encounters its benthos and it picks up its symbionts, maybe it picks up a variety of them, and over time it winnows down to a specific strain, and maybe that strain evolves with the coral. But also maybe there is some hybrid thing that has reseeded, and this is just a perpetuating cycle, and we don't really understand what's happening yet. Also, there could be environmental partitioning, of course. So if you are more likely to settle on the crust, maybe you pick up yellow versus in the mid-slope where you'd pick up red. And so maybe it's just a function of where they were sampled as to why we're seeing this pattern. And so yes, light, depth, and nutrient availability, those could all perhaps create some of these habitat preferences for the different symbionts. So work that my students now are going to do is kind of follow up on some of these scenarios that we've put out there to actually try to dig into this a little bit further of what's happening in this hybrid host. So one of the questions that we could get at is, are there preferred host symbiont pairings during symbiosis onset? And that we could do some lab choice assays. That's what we're planning to do. We're also going to try to pair that with some admixture mapping, so add some genomics to see if we can identify regions of the genome that might be more specific to particular pairings. We can also then do environmental field choice assays. So do the environment symbiont pools dictate the colonization dynamics? And we think that, if we put them to the nearest neighbor coral colony, we would assume that they would pick that up, but we don't really know. So we're going to put that out and see what happens. And finally, we do want to know whether or not these symbiont strains are evolving in their hybrid host. So we're going to try to rear some of these hybrids with a specific known inoculum of a symbiont, as well as the parental species, and see how they change over time. And that's the hardest one to do. All right, so the direction and frequency of hybridization varies across this range. We did find some evidence of genomic conflict in the F1s, and we think that that's probably why we're not seeing an influx of F2s. We do see symbiont differences that were observed by the host taxa, suggesting this onset of speciation, perhaps. We also saw the Esphitae were from the hybrids are more similar to Palmata. So again, we've got these different ideas of whether it's environmental partitioning, just a function of drift, evolution, and obviously it's probably a combination of all of those. So coming back to the kind of questions for this, was this an evolutionary dead end or an evolutionary rescue? I guess the thing is that we see back crosses, so they're not really dead ends in the sense that we can move adaptive alleles back into the parental population, but we think that those events are very rare because they're hard for us to get. I like surveyed 100 hybrids and I showed you 2 back crosses. So it's a lot of work to get more back crosses. Of course, laboratory experiments could get us there a little faster. So we think that we've identified some genomic incompatibilities that probably lead to this hybrid breakdown, and that's why we're not seeing later generation hybrids. But on the flip, maybe this isn't a bad thing because they're not going to overwhelm the parental populations, meaning that they're not going to overtake them with their genetics. So they're not going to mix up everything, and so we will still have 2 distinct species. So you can make a flip on that. But what I do think is that they can restore ecosystem function in the short term. So they can act as ecological rescue, but maybe not evolutionary rescue. And so we can put these out because they don't impose a big risk to the parental species. And they can also act as a refugia in case there are these novel symbionts that are adapting to these different hybrid microenvironments that we want to maintain. Okay, so from that work, that was a lot to go over. We took all of that genomic data and we thought, how can we take advantage of all of what we collected and turn that into something useful? So the lab that I was in was like the lab where all of the coral genotyping happened. And it was all microsatellite based and it took a lot of effort. And so we were trying to move that process out of the lab because we were tired of doing it, frankly. But also it's an important thing because if you look out on a reef, you're not going to know who's who just by looking at it. So the only way we can track individuals and understand the success of our restoration efforts is to actually get a genotype. So that motivated us to build a tool where we could track the genotypes. It had to be affordable to practitioners. That was really important. And that the practitioners didn't have to know any bioinformatics to run it. That was also very important because not everybody gets that training. We wanted to create a repository that was open to everyone, that wasn't just limited to the people who were using the tool. We wanted to be providing high resolution genome-wide information, not just 100 markers is sufficient to tell you who's who, but maybe we could actually use this in a long-term, more think about aquaculture or agricultural type applications where we want to understand traits and how they are related to their genotypes. And that, yes, the main thing was that it had to be open access. And so within the first, I should back up. So we developed a genotyping array, which seems kind of like old technology, but surprisingly in agriculture and in aquaculture, this is the standard practice. And so we developed an array with genotypes that were discriminatory for the different species, the different populations. We also put some symbiont probes on there so we could also identify the symbiont genotypes. Within the first 2 years, we were able to identify almost 1,500 genets out of the nearly 2,500 that were sampled. And this has been actually being used. I'm pretty sure at this point we have every Florida coral in the database, like every single one, because it's been adopted by the Mission Iconic Reefs, which is this large outplanting effort in the Florida Keys. And so this is just one way to show how we've been able to take this data and kind of transform how restoration is happening in a pretty short amount of time. The other thing that this highlighted pretty clearly to us is we need genetic management plans, because most restoration practitioners are putting out coral and they have no idea what they're doing. So we are really partnering with them to educate them on how important this is. And that's all I'm going to say about that. But if you have questions, happy to answer. Okay, so this last little bit is about how juvenile corals can inherit somatic mutations. And I want to point out my co-author on this was Kate Vasquez-Kuntz. She now works for Zymo. So earlier in this talk, I said that coral polyps are genetic clones of their neighbors or something like that. I fumbled my way through. But actually, in fact, each polyp has some degree of genetic mosaicism, meaning that there's some cells within a given polyp that may not be identical to its neighbor. And that's just through the process of somatic mutation. We also have that in our cells. And so they have slightly different genetic makeups. And there are two different sources of mutations. You can have germline mutations, which are in the blue here, and you can have somatic mutations, which are in the orange. If you have asexual reproduction, these mutations can actually rise to the point of fixation, meaning that that original little orange mutation has now become fixed in the neighbor. So it's not a mosaic, but at this point, all of the cells have the same fixed mutation. And so we took advantage of this fact in the way that we were trying to identify how mutations were being inherited, because they had to reach a certain threshold to be present. And from previous work, we know that adults accumulate these mutations, fixed mutations, and that they spread with linear growth and fragmentation so that they spread across the reef. And there's evidence that these mutations also show up in the gametes. And so here we really wanted to connect from that evidence that they were in the gametes into the offspring. And so that's what we sort of got at was, are somatic mutations inherited? So to do this, we, like I said earlier in the talk, we took advantage of the fact that we had the spontaneous development from eggs from a single colony. We called it the parent colony, which is right there in the center. And we were a little suspicious of this. So we also collected 5 neighboring colonies because we were like, well, maybe some gametes from one of those got in and that's why we're seeing this. And all of this happened before it got back to the lab. So there's like a little boat time. So this was all happening on the boat. Once they got back into the lab, some of those gametes, they did try to fertilize with Florida. So this was part of that assisted gene flow project we were talking about earlier. And so then they reared these in Florida for 4 months. And at that point, that's when we sampled them. But they were a mix. We didn't know who was who. So that was kind of fun. We extracted DNA from these 2 to 4 polyps each. And I just want to point out that the majority, when you do an extraction, is going to be somatic tissue and not germline tissue. So we're pretty confident that these were somatic mutations, and we did many things to kind of show that. We used our genotyping platform. We processed it with the standard workflow. And then we were able to identify these mutations as, again, these fixed mutations in the parents. I put quotes here, parents, because we looked at all of those as potential parents. And then we identified mutations. If we found them in the parents, we then looked in the offspring. And then we actually did a restriction fragment length polymorphism to confirm those because genotyping is messy and not always accurate. So we wanted to make sure that we were really seeing a true pattern. So the one thing I will first show you is that the neighbors were the same genotype as the parent. So they were all fragments of the original genotyped colony. So they were all the same. So we considered them all parents because we weren't really sure where the gametes came from, but they were all the same genotype. We also did see some level of within colony mosaicism. So here I'm showing you for the different individuals, we were able to track about 1.4% of these as mutations. Now, when we looked at the offspring, we looked at the dark bars here are the number of mutations, and then the light blue bars are the number inherited. So about 50% of those mutations were actually inherited in the offspring. And you can see that majority of the loss of heterozygosity mutations were inherited, but not the gain of heterozygosity mutations. And like I said, we did RFLP. So here is a beautiful gel image. And what I can walk you through here is the C means cut. So those are the ones where we gave the enzyme. The U means uncut. And so for colony 3, we found 2 offsprings that shared the same cut, meaning that they shared that same mutation. And for colony 5, it did not have that mutation. So we were able to confirm this in a handful of the SNPs that we identified as being inherited mutations. Okay, so to summarize, we were able to show transgenerational inheritance of genetic variation in coral offspring, and that this can bypass that germline barrier. The somatic mutations can also then be dispersed via the larvae, which could enhance genetic diversity across the geographic range. But the mechanisms of how this occurs is still unknown. And so here we put some ideas out there based on how other cnidarians do this. So you can have dedifferentiation or transdifferentiation, which just means a soma cell becomes a germ cell. And there's 2 different ways in which that can happen. So that's a little bit wild of an idea, but that is one way in which you could bypass the germline. The other idea out there is that there is never a fixed germline in cnidarians. And if there is, it develops really late in life. And so there's this time in life where you can bypass that. So mutations could pass that way. And so while we were doing this, there was another group that was also working on this, which is kind of reassuring. So they found similar things, but they proposed a different way in which these mutations can end up into the somatic cells. And that this is coming from a mutation in a stem cell, of which then is making its way into the somatic tissue line, into the germ line. So that's another study that kind of followed up a couple months after ours came out. So clearly working on it at the same time. And so that's just another way in which I don't have any stake in this. So take whichever hypothesis you like best. That's where I'll leave it. But there are multiple hypotheses about how this could happen. And it's open for people to continue to explore. So my lab now, I'm partnering with Dustin Kemp at the University of Alabama, Birmingham, and Bill Fitt at the University of Georgia. They have this collection where they've been going out for the last 20 years and sampling the same coral in the Florida Keys. And so they have a whole collection of coral samples from the same colony. And so what we're interested in looking at is how host mutation accumulation is changing across the last 20 years. We've seen instances of mass bleaching in the Florida Keys, so we can look at that over that span of time. And there has been one disease outbreak. But what I'm more excited about is trying to get at this symbiont mutation rate. And Kenny and I were talking about that. So that's kind of an exciting direction. So to sum up what I presented on all of that, hybridization can provide a novel source of adaptive alleles to the parental species through this process of backcrossing. but those are rare. And we think that later generations are just likely to not occur because of those genomic conflicts. We found evidence. Okay, sorry. We found evidence that a single symbiont species has adapted to its respective host taxa. And that may provide an alternative route to finding thermally tolerant symbiont strains from the hybrids that live in these more extreme environments. So thinking about another way in which we could use the hybrids in restoration, not only because they provide this ecosystem structure, but they also might provide some novel symbionts that are more thermally adapted. We also found a novel way for corals to adapt to by passing their mutations, whether they're neutral or beneficial, onto their offspring. And together, this data highlights the importance of tracking genetic diversity of both the coral and the algal partners to investigate the trajectory of these endangered species and to inform species management. Okay, so kind of back to this, hopefully I was able to demonstrate through a lot of population and comparative genomics how we're trying to work at conserving the species in that space. And this builds on some of the foundational work I've done more in experimental approaches using model systems. So this last little bit, I'm going to make a plug. I'm looking for students. So if you know of any students who are looking for a lab, specifically, I'm looking for students to come work in these model systems, Acceptasia and Cassiopeia. They both form symbiosis with the same symbiont as corals. So they're these good lab rats for studying these questions. And then you can balance Cassiopeia as the upside down jellyfish. So you can take a scyphozoan and compare it to an anthozoan and see how those processes have evolved. And so I'm really interested in lipids. This is something I worked on as a PhD student, and I'm kind of returning back to it. So I worked on this thing called the sphingosine rheostat, which is like a lot, but don't get too caught up in it. Basically, if you block this one enzyme, it leads to the reduction of this one lipid. And so what you can see, and I wish I had a pointer, the during host, kind of right in the middle or the C, if you give them this lipid blocker while they're trying to take up symbionts, you can effectively block onset of symbiosis. So they do not take up the symbionts. And it's at the same rate as dead symbionts. So that's how many symbionts they take up. And we did a couple other combinations, but that was the most compelling. So the next kind of direction I'm interested in is, are the symbiont-derived lipids playing a role in symbiont recognition and stable symbiosis? Kind of digging into this question a little bit further on these lipids. And with that, I'll thank my lab, which is very new and kind of up and coming. And then a lot of the work I presented today was from the time that I was in the Balms lab.